I would like to introduce to you some of the concepts and some of the questions that we are trying to address in human embryonic and pluripotent stem cells. And I will start out with giving you an introduction of why I think this is exciting and why this is a very helpful tool and why genome editing is a helpful way to make the best out of stem cells and to potentiate what stem cells can do to understand human disease. And then I will give you a few key experiments that serve as proof of concepts that we use to demonstrate what can be done in stem cells. And this is work that I have, as Dana said, done in Rudolf Jenish's lab. And then I will give you one example of where we use the Cas9 technology to interrogate one of the most frequent mutations in human cancer and how we use Cas9 to elucidate how this mutation is facilitating cancer. Okay, so with that summary of what I would like to cover today, this is the working paradigm of iPS disease modeling. The groundbreaking work of Yamanaka showed that you can take a somatic cell, and by the expression of just a few transcription factors, you can take this fully differentiated cell type and reprogram it into what is called a pluripotent cell, which has the ability to differentiate into all cell types of the human body. And this is a fundamentally important finding because this opens up the ability to interrogate diseases that are manifested by a genetic variation in the patient and allows us to derive the tissue that carries this specific genetic variation. And the idea is to use this to analyze what is the underlying principle or mechanism of the disease and conventional iPS disease modeling. Eventually, these cells that are differentiated could be used to derive high-throughput drug screening or to ask questions that are otherwise not directly addressed in conventional animal models. And the example that I like to give is in, for example, neurodegenerative diseases. The cell type that is affected by the disease is diagnosed in a patient where the cell that you actually want to study is lost. So the concept of how do we understand a disease like, for example, Parkinson's disease, and I will have one example of Parkinson's disease, how do we understand and derive models to understand this in the context of the human disease is very challenging because the cells is lost during the disease progression. So this experimental paradigm allows us to derive cells that are coming from a Parkinson patient, have the same genetic predisposition, and then derive neurons that, in the case of Parkinson's disease, dopaminergic neurons, are of human origin and can be used in disease modeling. And I think this is the fundamental idea of iPS disease modeling. The challenges of this idea are that, of course, we are all genetically different, and that if you isolate iPS cell from a patient, then the mutation that causes the disease in this patient is not the only mutation. It is also many other mutations are leading to genetic variability in between individual patient iPS cell lines. The other challenge of this idea is that for many of these differentiation protocols, you need a very specific cell type, and we are still lacking many of the differentiation protocols that are necessary to really lead to dopaminergic neurons that recapitulate a dopaminergic neuron that is in a 60-year-old patient. And so these differentiation paradigms are one of the key challenges. And then once you have this dopaminergic neuron from a Parkinson patient, you want to actually study this in the context of the disease. You want to manipulate it. And this is the state of, or was the state of iPS disease modeling and the challenges of iPS disease modeling. And when we started to use genome editing or establish genome editing, we thought that the developing genetic tools that allow us to overcome these challenges by manipulating the genome of iPS cells, we could facilitate this approach and this idea. So the problem at this time where we started this, and we started out addressing this first with conventional targeting approaches, there are a few reports by Thompson and Zwacker that show that you can actually achieve homologous recombination in human pluripotent stem cells, but the efficiency of these approaches are extremely low. 1 in a million cells, 1 in 10 million cells will undergo the homologous recombination event. And so this is not a feasible or tractable system that allows to generally manipulate cells. And the other thing is that you're all familiar in mouse genetics that we have expression locus like the ROSA26 or the collagen locus that allow overexpression systems in mouse cells. Similar genetic tools were not available at the time for iPS cells, and any random integration event into the genome would lead to strong variegation effects, methylation effects, and silencing effects that impeded general work with iPS cells. So we turned our attention to the use of site-specific nucleases, and I don't have to introduce to you site-specific nucleases in order to facilitate genomic editing of human pluripotent stem cells, with the idea that this then can be used for disease modeling. And I will refer to any of these nuclease platforms as site-specific nucleases because the take-home message from the studies that we have done over the last years is that once you have a double-stranded break, in first approximation, the events that happen afterwards are platform independent. So it doesn't matter if you have a zinc finger, a talon, or a Cas9. For a stem cell, the stem cell will deal with the cut very similarly. I will quickly review the process, however, because I came up with 3 very simple rules that if you follow them, you learned a lot about the different ways of how you edit in which organism. And I came up with a set of 3 rules that if you follow them, you will successfully edit in human pluripotent stem cells. And this is based on the mechanism that we think the repair event happens after a double-stranded break is generated by any of the nucleases of choice. You generate an overhang. This overhang will find, and I'm talking, sorry, I should mention, I will talk about homology-mediated editing. I will skip all of the NHEJ stuff because that is completely trivial and you guys know about that. Single-stranded overhang will invade a repair template that bears homology. This will then be used to synthesize DNA that is complementary to your repair template, and at some point it will generate DNA sequences that are homologous to the other side of the break. This can then be resolved by finding this homology of the other side. Flaps are trimmed, and gaps are filled in, leading to a repaired double-stranded break. Based on this principle, in human pluripotent stem cells, if you follow that you want to introduce any changes as close as possible to your double-stranded break, you're in very good shape. This is because the resection of your double-stranded break and the generation of your single-stranded overhang is very limited in human cells. And so you want to introduce any mutation within 20, 50 base pairs. This is probably very different in mouse cells. That's why I'm making the point of specifically human pluripotent stem cells. If you probably, if you want to make only, if you want to, you will probably only succeed by making modifications to one side of your double-stranded break unless you want to have several independent cutting events. So if you want to only have one introduction of a double-stranded break, you will most likely only introduce modifications that you incorporate on the left or the right side of your homologous repair template. And it is very important that you follow the rule of any insertion that your insert cannot have any regions of homology that are homologous to the left or the right side of the break, because it will allow your repair template to resolve the double-stranded break prematurely. And that is usually happens when you have something like 50 to 100 base pairs of homology. These are the 3 rules that I think are very important for human ES cells to successfully edit. And these are based on the experiments, or these findings are based on the experiments that I will go through. We used site-specific nucleases to provide proof of concepts that site-specific nucleases can facilitate gene knockouts by gene disruption, that they can be used to generate reporter cells, and we established a safe harbor locus that overcomes these challenges of variegation and silencing effects. And I will summarize those experiments to you because they will highlight a little bit of what you can do in ES cells with this technology, but they also illustrate the fundamental challenges and principles of this repair. So I will start out with talking about the establishment of the AAVS1 locus as a safe harbor locus. The AAVS1 locus is the integration site for adeno-associated viruses. It has been previously reported to be very frequently, integrations of adeno-associated viruses land in this locus, and it has very limited side effects. So you can homozygously integrate these viral sequences there, and it seems to have not a great impact. Based on these findings, we decided to use site-specific nucleases that cut at this AAVS1 locus. And we generated donor plasmids that bear homology to the left and the right side of this cut. And we did, in this experiment, a first comparison. We applied 2 targeting strategies. One is a targeting strategy that is referred to as a gene trap approach, where the selection cassette that is integrated into this locus in order to facilitate the isolation of targeted clones is driven by the gene itself, so through a splice acceptor that will pick up the message from exon 1. And we compared this by the integration of what is called an autonomous selection cassette, where the selection is driven from its own promoter and then polyadenylated within the intron of this gene. And we found that if we co-electroporate, all of the experiments that I'm talking today about were done by co-electroporation of cells, with any form of in this case, the zinc finger nuclease with the donor plasmid, and then assaying how many cells gain in this case, the puromycin's marker, and scoring for what is the frequency of correctly targeted or edited clones compared to not correctly targeted clones. And what we found is that in this very first experiment, we found that 50% of all puromycin-resistant clones were correctly targeted, independently of if they were targeted by an autonomous or by a gene trap approach. And that was very encouraging. Finding this high targeting efficiency is compared to 1 in a million from the previous report by Zwacker that targeted the OCT4 locus, was a major step forward. We did a few very important confirmations. We showed that these human pluripotent stem cells are not grossly affected in their ability to maintain pluripotency. That is, of course, a concern because you're expressing a nuclease that could be genotoxic. It did not have an effect on the expression of pluripotency markers, as indicated here by this immunofluorescence staining. And we confirmed that these targeting events are correctly targeted using Southern blot strategy. I will come back to the Southern blot strategy in a moment because I think it's a very important point to make that the great advantage of editing in human pluripotent cells is that you can go through a single cell step, which means that we can work with clonal events, and we can, after the editing, claim that the cells that come out of a targeting event are isogenic. And every cell that comes out of the targeting event is genetically the same, which also puts a burden on the fact that you have to show that they are genetically what they are supposed to do, and we usually do that by Southern blot. So this was a very encouraging experiment. This was done in human ES cells. As a side note, I strongly believe that human ES cells and human-induced pluripotent stem cells are completely equivalent. we see, at least in terms of targeting, no differences in the ability to edit human embryonic stem cells or induce pluripotent stem cells. Here is a direct comparison of where we edited, let me see. Oh, sorry, this is the splice acceptor pure, where we did the same experiment again, where we targeted iPS cells, and we find virtually the same targeting efficiency. So the take-home message here is human ES cells and human iPS cells are the same. After this puromycin integration experiment, we decided, okay, this puromycin is not a helpful transgene. We decided to check for the integration of a GFP into the AAVS1 locus. But to do that, we extended this targeting strategy, having now a GFP expressed from an external promoter, in this case a CAX promoter, which is a very strong stem cell driving promoter. And we made a very important observation. We made the observation that targeting a locus in human ES cell results in a uniform expression of this exogenous cassette. All cells express to equal or very similar amounts, indicating that there are very few variegation effects, very few silencing effects. And then if we differentiate these human embryonic stem cells by an assay that is called a teratoma formation assay, you take these human ES cells that are targeted, inject them subcutaneously into the skin of mice, where they differentiate into all of these tissues. And then we performed immunohistochemistry. Everywhere where you see brown is GFP expression. We find that this GFP expression is maintained throughout the teratoma and throughout many of the human tissues, illustrating that this locus is really useful because it not only allows to express transgenes in human ES cells, but also in the cells that are differentiated from these cells. We extended the usefulness of this locus by showing that you can turn this into an inducible expression system where now the GFP in this case is driven by an inducible promoter that is dependent on the reverse transactivator. And we could show that this is a FACS analysis of human ES cells that after the targeting were treated with different amounts of doxycycline, which will render the promoter active. We could show a dose-dependent increase of GFP, showing that this locus that we can use control of this transgene. So with this, we felt very comfortable about this AAVS1. We made one further improvement. In this case, this promoter is dependent on the reverse transactivator. In this experiment, we showed that within a single targeting step, you can trans-heterozygous human ES cells. Within one targeting step, you can target one allele of the AAVS1 locus, in this case with a fusion protein of GFP to H2 to a histone variant. And on the other allele, the M2R-TTA, which will render this promoter active under the addition of doxycycline. And if you do that in a human ES cell, you get something, human ES cells undergoing mitosis in real life, and you can visualize that under live cell imaging. This is a human ES cell colony where every now and then you see a cell dividing, and these are still images. From an editing point of view, this is interesting because this is one electroporation step where you generated a human ES cell that can be triggered to express a gene of interest. From the day that you decide that you want to have this expression line to the time where you can do your first movie is about 3 weeks in human ES cells. If you had to target this sequentially, then it would take 6 to 8 weeks. Okay, so these are technicalities of the AAVS1 locus. But it turns out that this AAVS1 locus is frequently used to actually achieve exactly that. If you want to express something in human ES cells, this is a locus to go for. It will have very robust expression. I want to quickly now talk about the AAVS1 locus. This AAVS1 locus was one of the first that we investigated, and we wanted to figure out how specific is this. How specific are the nucleases that we are using? And I want to quickly introduce to you this assay, which is called CELEX. sequencing where you determine the specificity of a nuclease based on an in vitro assay where you ask what is the nuclease actually binding to. And these are, here we compared the specificity of AVS1 targeting zinc finger nucleases, and in this case it's TALEN nucleases that have approximately the same binding site, and the assay is done by asking, are these, when you have these proteins as recombinant proteins, what is the actual DNA binding sequence that they're going to interact with. And this assay, I think the reason why I have this slide up is because it shows that these are highly specific nucleases. Every time that you see a bar above this line, it is the intended binding and the correct recognition of the nuclease that it's intended to in its recognition sequence. Every time you see something below the bar, it is the binding to a base that it is not expected to bind to. And the conclusion from these analysis was that these are highly specific. The point why I'm bringing this up is this leads to the question of prediction of off-targets. We did some efforts to figuring out assays of how to use this prediction to survey where are potential binding sites within the stem cell genome and interrogate if other sites other than the AAVS1 are cut when you express these nucleases in human ES cells. And here is a case for where we did this in, where we predicted the potential binding of these nucleases and then interrogated those sites by using an assay that is the cell1 assay, which I'm sure you guys discussed. And the take-home message is that most of the nucleases that we're working with are highly specific. And in the context of human iPS disease modeling, I'm telling my students usually that they should not worry too much about off-targets. If you don't want to bring something into the clinic, it is probably a faster and more prudent way to use two Cas9s that have different PAMs and different recognition sites to knock out a gene and confirm that the phenotypes are the same, rather than going through an elaborate off-target analysis where you probably do not saturate any of the potential off-targets anyways. If you have a knockout, you complement your phenotype, and you are probably pretty well off, okay? So this is my personal opinion, but I think that off-targets in the context of disease modeling, if you design your experiments smart, are not the biggest concern. Okay, so I told you a little bit about this AAVS1 locus, then diverge a little bit into specificity of nucleases. Now I want to talk to you about that we have edited quite a few loci, and the question that very often comes up, is there a correlation between if a gene is active or not active in the efficiency of if it can target? And we try to address that by targeting a gene in human ES cells that is not expressed, which is this gene PITX3. And it follows the general theme of Parkinson's disease. It is a transcription factor that becomes transcriptionally activated when human ES cells or when cells differentiate into dopaminergic neurons. And so we tested the efficiency of nucleases that cut in the first exon of this PITX3 gene, and then assayed the efficiency with which this GFP is integrated if we use a donor plasmid that bears homology to the site of the cut. And what we find is that this targeting has similar efficiencies to targetings that are done in genes that are highly expressed in human ES cells. So a non-expressed gene is not prohibitory to being edited. At the time, this was actually quite important because it was not clear what the relationship between the transcriptional status is. And I still think that there might be a correlation, but there is no convincing data that would argue that at least the transcriptional status, I'm not talking about the heterochromatic status, is directly related to targeting efficiency. This is also a very insightful targeting for us because we learned something else out of this. And this is, I'm here showing you the main figure, southern blot, where the top band is the wild-type band, and there we have the targeted band. This is the supplemental figure. And in this supplemental figure, you see that the top band is the targeted band, the bottom band is the top band is the wild-type band, the bottom band is the targeted band. Here is a nice homozygously target-edited human ES cell clone. Only the targeted band is there. And this is checked with two independent probes, a 5' probe and a 3' probe. If we had done this, analyzed this only with the 5' external probe, then we would have probably called this also a homozygously targeted clone. However, if you look at the homozygous targeted clone here, on the 3' external, you see an additional band. Okay? And that is something that we refer to as an aberrant targeting, a targeting that has an additional integration, and that you would not pick up if you analyze this clone just by PCR genotyping, right? Because the correctly targeted alleles are there, and you would probably span homology arm internal to the GFP on one side and say, oh, everything is hunky-dory. You might even sequence your PCR product, and it's completely fine on one side of your integration event. And that goes back to one of the comments earlier. You can have heterologous integration events or tandem integrations, or many events can happen that look okay by PCR but are not by Southern blot. And so the gold standard should be Southern blot analysis if you have the ability to go through a clonal targeting step. I'm highlighting this here we have a pretty good idea of what some of these events are, and we hunted many of them down to actually learn what they are. And I'm more than happy to discuss that with you later. Here I'm just highlighting how often you find events that we have to call aberrant events, despite the fact that they are also correctly targeted in the correct locus. And this can be substantial. This can be 50% of all integrations can be corrupted this way. And right, so very high numbers of additional integration are seen in human ES cells, and it's a disclosure and a warning to anybody who does this that you should always check for it. Okay, so the take-home message here are twofold. One is non-expressed genes can be targeted, and it is important to correctly genotype your cells. This experiment that I'm going to tell you about now is an experiment that I didn't do myself. This was done by a colleague of mine, Frank Saltner, and he decided to tackle a question that I find super important. It is this problem that I raised earlier, that iPS cells are genetically heterogeneous, and if you want to appropriately disease model and attribute a phenotype to a specific mutation, this should be done in an isogenic setting. And this is described in this overview figure that Frank shown. Is he took a patient fibroblast and reprogrammed them into patient-derived human-induced pluripotent stem cells. They carried a mutation in the alpha-synuclein gene that predisposes to familial forms of Parkinson's disease. And then he used site-specific nucleases to take this mutation and repair it into the wild-type form. And that's resulted in what we refer to as isogenic cell lines. They're isogenic except for this particular mutation. And then he took these cells and differentiated them into neurons, again into dopaminergic neurons. This conceptually, I think, is currently the state-of-the-art way of doing iPS disease modeling, because if you find something in this setting, you will be able to, with very high probability, be able to confirm that this phenotype is related to this mutation. The alternative approach that he also demonstrated in his study is that you can take wild-type ES cells or iPS cells and introduce the mutation, and you will generate, again, an isogenic set of isogenic cells. This is the way that this experiment was done. This is the alpha-synuclein locus, and this is the position where this genetic variation occurs. And he used, in this particular experiment, he used either double-stranded homology donors or single-stranded nucleotides to either insert this mutation into human ES cells or to correct this mutation in human-derived iPS cells. And these mutations are occurring in a heterozygous configuration in the patient. And so this is just a read of where he took wild-type human ES cells and inserted this mutation. And then he took these cells and differentiated them into dopaminergic neurons. And this cellular system has now been used in several studies to really find the underpinning molecular mechanism of how this alpha-synuclein variant leads to Parkinson's disease. And I think that Susan Lindquist even has a patent on using these cell lines to has identified small molecules that can alleviate phenotypes that are found in the system. I think this is it's really an elegant way of showing the power of the system, because this brings back to nobody has ever had, prior to this study, any dopaminergic neurons that carried this variant, right? So I think that's exciting. Okay, I'm going to skip through some of these. We did additional work where we showed that you can target OCT4, which is one of these pluripotency genes that Yamanaka used to reprogram cells. We made what is called a lineage reporter for pluripotent stem cells, and where we integrated a GFP at the last amino acid of this OCT4 gene. And we did this in a way that the only modification in the pluripotent stem cell genome is really just the insertion of this GFP at this position. We did this by first having a selection cassette in there that then was removed by CRE-mediated excision. This targeting, again, was very efficient and resulted in OCT4 positive cells. If you take these OCT4 positive cells, we are growing our human pluripotent stem cells on feeders. So there is a population of mouse embryonic fibroblasts that are not OCT4 positive. But if you take these OCT4 positive human ES cells that are targeted in this way and differentiate them into fibroblasts, you lose this OCT4 expression. And it has been used for then looking at the transcriptional regulation of OCT4. The point that I want to make in this slide is that you can have several genetic steps at the same locus in order to achieve your correctly edited cell line, where you start out with having an insertion, and then, for example, here you remove a cassette. By now, we have also made several iterations using first the Cas9 at one locus and then recutting with Cas9 on the edited allele to make a second modification. And so this is all possible. Okay, with this introduction, I want to get into a little bit of a targeting kind of, let's see if you guys follow me. I like this experiment because this experiment is again targeting this OCT4 locus. And we compare two different things here. We compare what happens if we violate one of the rules that I told you earlier, which is you want to make your modification as close as possible to the cut that you can. And we did this by testing zinc finger nucleases that cut after the first exon of OCT4. And then we derived two different sets of donor plasmids. One is a donor plasmid where the homology arm starts directly at the side of the double-stranded break. And the other one is a little bit off by 106 bases, and where the homology arm starts at the last amino acid of this exon. And we compare what happens if you drive this GFP under a selection that is spaced by this self-cleaving peptide and a puromycin. So it's a gene trap. Only cells that have this GFP puro-integrated will be puromycin resistant, versus a GFP that is directly fused to exon 1 and then drive the puromycin through the self-cleaving peptide. So we are asking in this experiment, this is our positive control, we are asking here, can we remove, by this strategy, 106 bases? And I wouldn't tell you if this didn't work. This is from mechanistically at ask, are there clonal events that after the cut generate enough single-stranded DNA so that we can generate a homologous pairing in exon 1 that then will use this to generate a GFP. And we generated these clones. I found this at the time very exciting because it means that with some frequency these events are happening, and if you have a strategy that can generate those, you will find this particular clone. By now this might be pretty much standard, but I still think that understanding the principle of this difference is important for when you design your strategies. Okay, so at the end of all of this, we were at a point where we could use site-specific nucleases to facilitate this in vitro disease modeling, and I think we now have all the tools to actually do that. We can achieve overexpression, we can do point mutation repair, we can generate lineage reporter that report on the differentiation status of human ES cells. Okay, so I would like to introduce to you now an experiment where we actually applied this and where we used this. And of course, we switched to Cas9 because Cas9 all of a sudden made things so much easier. I mean, stuff was, you can give a talk and make it sound easy, and then Cas9 comes around and it actually becomes easy. So I introduced to you this question of what is IPS disease modeling good for? And when I started my own lab, I decided that this is actually a really expensive, elaborate, and convoluted way of studying human disease. And I would not suggest to any of you to use human pluripotent stem cells for disease modeling if you can answer your question in an animal model that runs around or swims around. If you can address your phenotype in something that is alive, go for it. Okay? So when I started my lab, I decided I'm going to study something where I'm trained in this and I know how to do this, where this actually is needed and where you can only address this question in the context of human disease. And luckily, when I started my lab, there was something that just popped up that I got really interested in. And that is the question of, it's a cancer question, and it is a report that showed that in the human genome, the most frequent non-coding mutation is a mutation in the telomerase promoter. Telomerase, I will introduce that to you, and I will introduce to you why this question can only be answered in human cells. So what was reported in 2013 is that in the gene that expresses this gene TERT, which is the protein that makes telomerase, is the most frequent mutation in cancer. And this mutation is found in a very specific spectrum of tumors. So it's found in melanoma, it's found in lipoma and bladder cancer, but it's, for example, never or very, very rarely found in lung cancer, very rarely found in any blood cancers or intestinal cancers. This specific non-coding mutation, 120 base pairs upstream of the ATG, generates an ETS transcription factor binding site that a few weeks ago was reported to be a GBP alpha mediated transcription factor binding site. However, this is a standard GWAS found report. Okay, this is highly prevalent, and it's clearly that it has to have some meaning, but it is a GWAS study, so it's not clear how this is functionally related. And so we set out to address what this most frequent non-coding mutation in cancer does. Okay, so to put this a little bit into the context, we now have a GWAS identified mutation in a promoter of a gene that I have to still introduce to you. And I will do that by explaining to you what telomeres are. Telomeres are the end of linear eukaryotic chromosomes. Every of our chromosomes end in a repetitive sequence that is TTAGGG. And this is about 10 kb on every chromosome end in human cells. And this structure, this DNA sequence at the chromosome end, provides two main functions in cells. For one, it mediates that terminal sequence loss by nucleases or by the end replication problem. so by eating up your chromosome end every time that you divide, is not leading to sequence loss of genomic information. And the second thing that telomeres are doing is they make sure that the natural chromosome end is not recognized as an internal break. For example, if you have a Cas9 cutting internally in the genome, this DNA breaks are very fast repaired. We know that. Telomeres are also a DNA end, but they are not repaired. And this specific difference between the two ends is one of the functions of telomeres. And so the sequence is de novo synthesized by this enzyme telomerase that has this protein component TERT and has an RNA component that has a sequence that is, has a template sequence to generate these TTA GGG repeats. Okay, why do we have to address this system of telomerase and promoter mutations in telomerase in human cells? This shows the difference. Mouse cells have telomeres that are excessively long compared to human cells. They have telomeres that are 10-fold longer than, 5 to 10-fold longer than we find in human cells. And in mouse cells, telomere length is constant throughout every somatic tissue. And it doesn't change. This is in striking contrast to human cells, where in human embryonic stem cells, telomeres are shorter and are maintained over time. However, when a human ES cell differentiates, telomerase is transcriptionally silenced. This protein TERT is transcriptionally downregulated, which leads to the fact that telomeres shorten progressively with every cell division. And at some point, then this leads to a proliferative stop. The chromosome end is sensed as a site of DNA damage, and cells will arrest. This is the conventional Hayflick limit that human somatic cells engage. This doesn't happen in mouse cells because of TERT not being transcriptionally silenced when cells differentiate. And at this point where normally cells would divide, the immortal phenotype of 90% of all tumor cells depend on the reactivation and restabilization of telomere length through the activation of telomerase. And so we wanted to put this experimental or this knowledge into the context of these TERT promoter mutations, because one of the questions is, these TERT promoter mutations, are they sufficient to promote this process? And that brings us to the key questions that we wanted to address using Cas9-mediated genome editing. We wanted to ask, what is the effect of these TERT promoter mutations that are really just 120 nucleotides upstream of the ATG? Are these mutations sufficient to mediate the step of counteracting telomere shortening when cells become tumor cells? Or do you need additional changes in the cancer genome? In the melanocyte, these are one of the most genomically unstable cancers that are around. Do you need selection for any of those additional changes? Why are all these TERT promoter mutations within this particular region? Why are there no upstream? Why these particular mutations? And then why are they in a specific tumor subset? And we addressed this using genome editing. I don't have a slide for that, but we did something that a student of mine started out conventionally trying to edit every of these mutations. And he made Cas9s, and until we learned that it's actually really difficult to generate Cas9s that cut within this promoter element of TERT, because it's very GC-rich. This is it's a standard GC-rich, non-TATA containing promoter. And the first 600 base pairs of this promoter have 80% GC content. So we failed to actually conventionally introduce the TERT promoter mutations into human ES cells, and we failed to generate isogenic cells by just cutting at the site and then having a repair template that would introduce that mutation. So we did a trick. We decided, okay, we can't deal with these 600 base pairs that are just GC by genome editing. So we cut out a larger chunk. We cut out 2 kb, and we did this by having 2 Cas9s that cut at the site of these 2 kb, and we generated homozygous deletion cell lines that had this sequence lost. And then we added back this sequence, but we added it back either in a way that it contained any of these TERT promoter mutations or didn't. And with this two-step approach, we again generated isogenic cell lines that differed exclusively at these mutation sites in the TERT promoter, that either we added back the wild-type allele into the promoter or contained these 3 specific point mutations. And then we asked the very simple question, what do these promoter mutations do to the regulation of the gene that is downstream to TERT? And there was the first surprise, because these 3 mutations occur in tumors. However, this mutation 124 bases upstream is the most frequent mutation. And this mutation is the only one out of these cancer mutations that actually resulted in a transcriptional upregulation of TERT. The other 2 did not. And here are a few controls for this, or is the control that these cells after the editing remain pluripotent, and that this is not an effect of differentiation, that these mutations introduce differentiation. And here I show you that the average of these individually derived isogenic clones that came from a single targeting event. So these are independent clones that I show you here. So that was a surprise, where there was no reason why only one of the mutations should reveal a phenotype in these cells when all of them are associated with cancer. We said, okay, maybe there is something that we can't really address in human ES cells, because in human ES cells, telomerase is high. So in these cells, telomere maintenance is actually occurring, while these mutations are selected for under conditions where telomeres are short and where telomerase becomes limiting. So we decided to take human ES cells that carried these mutations and all of these 4 lines and differentiate them into telomerase-negative cells. And we did this by differentiating them into what is called an embryoid body. Embryoid bodies are when you take human ES cells and culture them in suspension culture, where they will normally rapidly downregulate OCT4 and telomerase, so stem cell markers, while induce differentiation markers such as collagen. So it's a standard differentiation protocol. And what we found there is that indeed, I'm sorry that this is so low resolution, is that wild-type cells downregulated TERT transcription very efficiently in this differentiation protocol. This is the blue and the black line here. And in this case now, we find that all 3 TERT promoter mutation cell lines that only have these mutations fail to transcriptionally silence telomerase to the same extent as wild-type cells. This is not caused by failure of differentiation. Here we have the downregulation of OCT4, so all of these cell lines are equally differentiating and downregulating the expression of OCT4, and all of them equally induce the expression of collagen. And here is a normalization. When we normalize this to wild-type cells, at any of the given time point, we see that all of these mutations lead to a 4- to 5-fold increase of TERT expression in these cells. These embryoid bodies can then be continued to differentiate into fibroblast-like cells. These are in a wild-type setting completely telomerase negative, but in the case of these mutations, we have residual activity of TERT expression of about 10%. And again, OCT4 is completely silenced, further supporting this idea that these TERT promoter mutations are sufficient to transcriptionally upregulate TERT to a level where, to an aberrant high level. So this TERT transcription is only the generation of mRNA that then will lead to the expression of this protein TERT, but it does not tell us anything about the functional readout of this. And here we performed an enzymatic assay that asked for, is this increase of TERT expression also functionally related to the activity of telomerase, so the fully active enzyme? And we find that this is actually the case. This is an assay where you take a radioactively labeled oligo, and you ask in an extract for with what frequency and how actively does in this extract you get TTA-GGG, so the telomeric repeat addition to this oligo. And what we find here is that in these fibroblasts, that have only minimal TERT expression, you find very little activity, while we find very robust activity in the cells that contain these TERT promoter mutations. Okay, so we decided that this is very exciting because it could mean that these TERT promoter mutations are actually sufficient to lead to telomerase levels that are sufficient to immortalize cells, and I will show you an experiment about that in a second. But before we did that, we wanted to make sure that this is not just related to this one differentiation protocol. And so we decided to follow up on this by differentiating these TERT promoter mutation containing human pluripotent stem cells by differentiating them into neuronal precursors, which are abbreviated by NPCs. And then these neuronal precursors, which have low levels of telomerase activity, they are a precursor cell type or an adult stem cell-like cell type, can be further differentiated into neurons that are then fully telomerase negative. And when we do this as an alternative conformation, then we find that in neuronal precursors, wild-type cells have, compared to human ES cells, some residual levels of TERT expression. However, we find very high and very robust expression of the TERT promoter mutations in these neuronal precursors. And if we then differentiate these neuronal precursors further into new N-TUJ positive neurons, which are markers for mature neurons, we find that even in these cells, the TERT promoter mutations confer TERT expression and aberrant high TERT expression compared to wild-type cells. You can put these neurons on mitotic inhibitors, which stops the proliferation of these cells, and even under those conditions, we find some residual TERT expression. This translates again, even in neuronal precursors and in neurons, in aberrant activity of telomerase. So post-mitotic neurons that carry TERT promoter mutations have telomerase activity, which is absolutely absent in wild-type cells. To put this into the context of cancer and cancer progression, we ask, is the levels of telomerase activity that we see in these stem cells relevant for tumorigenesis? And we did this by comparing these cell lines that carry these mutations that are shown here in a titration where you assay really semi-quantitatively how much activity is there, and compared it to known, fully transformed, immortal cell lines that are around for 30 years. And we find that under these conditions, HeLa S3, for example, has telomerase activities that are similar to those found if we introduce this most prevalent cancer mutation. And even in fibroblast lines, we find that the activity is very high and is at least going to postpone the onset of telomere shortening to a point where we expected that these cells are immortal or will be sufficient to form a tumor. The full readout of telomerase activity is, of course, telomere length. And telomere length is assayed by looking at a southern blot that measures the terminal restriction fragment where you cut away genomic DNA. The only thing that is left over is your TTA GGG track. You run that out on an agarose gel, and then you hybridize with a radioactive probe, asking for what is the average length of your telomeres. And if we do this in human ES cells, we find that the TERT promoter mutations only confer a very slight increase in telomere length. This is expected because the TERT levels are only slightly increased in those cells compared to wild-type cells. However, if we then look at neuronal precursors or at fibroblast cell lines, we find that there is a pronounced difference in telomere length when you look at wild-type neuronal precursors that have only low telomerase activities compared to the telomere length of any of the cell lines that carry the TERT promoter mutations. And to put this into a little bit more of an in vivo context even, we took, again, these human ES cells and injected them into what I introduced to you earlier, this teratoma formation assay. Human ES cells subcutaneously injected into mice where they differentiate into all cell types. And then in this tumor that is growing in the mouse for about 8 to 10 weeks, assayed how long are telomeres. And again, we find that telomeres in wild-type teratomas are short compared to any of the teratomas that derive from cells that carry these TERT promoter mutations. And I think that this is highly relevant for cancer because it means that a single non-coding mutation in the promoter of TERT is highly likely to immortalize human cells. And if you look by I introduced this very conservatively by saying this is the most frequent non-coding mutation in human cancer. If you go just by base, if you just ask for how frequent is this mutation in any cancer, then you find that in melanoma, it's found in 50% of all melanomas. It's found in 60% of all bladder cancer. And if you sum up all cancers and then ask for just this 124 mutation, it's probably the most frequent mutation in all cancers. Because P53, which is the runner-up, is mutated in a variety of different positions. But 124 in the TERT promoter, just by base count, will be the most frequent mutation. Over all cancers, it's about 20%. And with that, that's my story.